Hey, Aaron, welcome to it's episode 34 of the Octopodcast. How you doing this month?
I'm good. How are you, Tony?
I'm doing well, doing well. It's funny, I think we were going to record it last week, or I don't remember if it was the week before. I think it was last week, and we wanted to let the news bake a little more, see if there was some more good cephalopod stories that we could share. And I think we got that. I think we have a pretty good list of stories to share this week. Let's jump right into it. I'll kick it off. Plans for the world's first Octopus Megafarm have been scrapped after years of backlash. So they've decided to pull their request to start a farm, which I think is a victory for cephalopods for sure. IFL Science carried this story, and basically said, we read a couple of quotes from it. The question of octopus aquaculture is riddled with ethical and environmental worries. Campaigners from animal welfare and environmental groups, NGOs, scientists, and the public have spent the last 5 years vocally objecting to the project. So reports suggested the amount of fish required to feed the octopuses was expected to be around 28,000 tons, or 2.1 billion wild-caught fish in its first year. So that is, very impactful to ecosystems and supply chains and, everything in between.
Yeah, I mean, it sounds like the number of fish required to sustain this mega farm would have required a fish farm to be set up next door.
Yeah, some kind of a racket going on here, I think.
Yeah.
So personally, I don't eat cephalopods. Do you eat cephalopods?
I don't know.
Yeah, and is it because you work so closely with them? Is that kind of...
Yeah. It's gotten to the point now, because I've worked so closely with them, that it's like, oh, eating a dog or a cat. Like, it's very familiar.
Samesies, I'm the same. Totally agreed. So for folks like us and folks in the cephalopod community, that's a good news story. All right, so that's our first story to bring you this month. What else do we have, Aaron?
Well, the next one I think might kind of tie into this a little bit. It is to do with cephalopod aquaculture, but I think that it's more from a scientific aquaculture and research rather than farming for food. So this was an article about genotyping in cephalopod hatchlings to determine their sex within 3 hours of them hatching. Now, this is particularly interesting because when we think of sex in individuals, I assume that many of us would come at it from a mammalian perspective. When humans are born, we're pretty certain if they're male or female. And that's just not the case with a lot of non-mammalian animals, and particularly invertebrates. So when cephalopods hatch, usually you have to wait until they've reached maturation or adulthood before they even start to develop what we would call, adult sexual characteristics. It's not readily visible when they're hatchlings. And for some cephalopods, those features don't even develop, and you have to dissect the specimen to determine if it's male or female. So this is really interesting because it is a non-invasive means of finding out pretty much immediately if it's a male or female cephalopod that has hatched. Now, this is important for aquaculture studies because very often science labs will develop a breeding culture of whatever species they're working on, so that they don't have to go into the wild and catch more animals to do the research on. Very similar to farming octopus for food. And they need to be able to keep a balance of males and females within this community so that they can continue the breeding population. You don't want to run out of males and you don't want to run out of females. So what this does is, by taking a swab of the hatchling, now this is very, very scientific into the genetic end of things, which is not my forte in any way, shape, or form. But reading through the article, it seemed like they had developed specific primers which latched onto a certain section of the cephalopod DNA, which, when amplified, if there's a 1-to-1 ratio, you have a male, and if it's not a 1-to-1 ratio of these DNA primers or these DNA sections, it's a female. And so they've created a primer which marks these. Now, for those of you that understand genetics, this probably makes sense. For the rest of us, it's pretty much like taking a highlighter and highlighting a portion of DNA and going, yes, this is the important stuff here. And then the machine just kind of continues to work on that highlighted section. And so by taking a swab of the hatchling and putting it through the PCR machine and amplifying this section, they can determine if they have a male or female specimen. So this is really cool. And it's something that I think will enable future science teams to be able to maintain these cephalopod communities that they have within their labs. And again, not have to go into the wild for more animals, which we like. We like not having to take animals from their homes. From a scientific perspective, future uses of this, recently there's been a lot of work on something called eDNA. So when we send submersibles or remotely operated submersible vehicles down into the depths, they're using water filter things that look for biological material in the water, and they filter through and use this eDNA to try and determine what species have been in the area recently. So it would be really interesting if this could be extended into that, so that we would know, hey, this thing that we're seeing on the ROV footage, we can tell what species it is because we've done eDNA. We can also tell that it's a male because we've amplified that area of like that. I think that would be really interesting if science could head in that direction.
Oh yeah. So that is a breakthrough of sorts. Very cool. And hopefully in a future, Aaron, maybe when we're doing this 5 or 10 or 20 years down the road, we'll say, hey, remember when this technology came out? Well, now look at us. Aaron, what else do we have in the news?
Well, connected to egg masses and hatchlings, we have a photograph of an Arctic bobtail squid. Now that in and itself is exciting. If we've talked about bobtail squid before, we've probably talked about the more tropical ones, which have very frequently been seen or witnessed burying themselves in the sand, and they kind of shuffle around and then push the sand over their head, and they're really cute. Love a good bobtail squid. However, this is one that we don't see too often, and that's mainly because we don't get to do quite as many research trips into the polar regions. But this is the Arctic bobtail squid. It's a species from the genus Rosia. And what was really exciting about this was not only that we got some video footage of it, and some photos of it, but that it was also carrying around an egg mass. So this was us witnessing this Rosia egg mass in the wild, and so that was really exciting for the scientists that were aboard the vessel. The article itself kind of goes over a little bit about bobtail squid and about Arctic research, but there is a really cute video there of the little Arctic bobtail squid for those that want to go and watch it.
Yeah, super cool. And this was through use of Yachts for Science. So there's this organization called Yachts for Science. Through this organization, research teams can use private yachts for shorter, specialized research trips to lesser explored marine ecosystems. So that's really interesting. I love that. It's almost like an Airbnb in the form of a yacht for scientists to get to remote areas and do some research. And the yacht owner, I guess, can make a buck. If you own a yacht, do you really need to make a buck? Maybe.
I mean, if you're really passionate about science, why not?
If I had a yacht, I would happily. Maybe I'd do it for free. But that's very cool. And so that's never been seen in the Arctic. Is that correct?
So we have, as far as I could tell from the article, we know about this species. It's not a new discovery, but it is the first time that we've gotten footage of it live in its natural habitat.
Yeah, and brooding for sure, right? Very
And
cool.
brooding, which is always exciting. I mean, with the vastness of the ocean, anytime that we see a cephalopod brooding eggs, it's like, oh, well, this is exciting, behavior and whatnot.
Yeah. So we went from aquaculture and raising cephalopods in farms to studying them in the lab to determine male or female, and there's kind of a connection there as we were talking about. And now we have this kind of connection here with a brooding bobtail squid, clearly female.
Yeah.
What is the next story, Aaron? Is there another segue here, or are we just jumping to a new topic?
There is not. We're jumping to a new topic. I thought we were doing really well with the narrative. This next article that I thought was particularly interesting was about brain size in cephalopods. Now, Tony, you and I both know cephalopods, very intelligent. And as an invertebrate, they've kind of had this convergent, parallel evolution to vertebrates where they've developed larger brains and photographic eyes and all of these very specialist features that a lot of other invertebrates don't have. And so this article is called Ecological, Not Social Factors Explain the Brain Size in Cephalopods. And I'm going to be honest, when I read the title, I kind of rolled my eyes. And I'll tell you why. It's because as a cephalopod researcher, when you go to deep sea conventions or to kayak or something like that, there's always talks on cognitive ability and spatial awareness and that. So in the Ceph community, we're kind of like, oh yes, it seems like environmental factors would be the big thing that would kind of help with developing cognition within cephalopods, specifically, I guess, octopus.
Yeah.
So when I read the title, I kind of thought, well, yes, duh. But then I went and I read through the article in a lot more detail. And I got to say, it's a deep article. It's got some really kind of big philosophical terms. There's a lot of abbreviations. It uses a lot of very complex mathematical formulas. And so my head really kind of started to boggle when I was reading it. But I got to a summary. And what it does is it looks at a whole bunch of different animal behavioral concepts. And for mammals, for instance, our brains are thought to have developed because of social connections that we've developed, social groups that we lived in and how we interacted with each other. And that's why we had to have brains that work particularly well. And the article itself makes a point of saying cephalopods, for the most part, don't do this. They live in isolation very frequently. They live in environments where they don't come across other cephalopods. And when they do, it's very minimal interactions. There's a few exceptions. I mean, cuttlefish would be a good exception to that.
Yeah.
And so they were looking through this and trying to figure out ways in which the cephalopod brain developed in such
Isolation.
interesting way.
Yeah. Mm-hmm.
Yeah. And so they came up with a scheme and went through research done on many different types of cephalopods. And in pretty much every instance, they found that the environment was the main factor that drove that cephalopod brain to develop. Even in groups where they do have social communication and they use color change and whatnot, that was not the dominant factor. Their mathematical equations came up with that the environment was still going to be the dominant factor. So, I guess in a way, I'm still going, well, yes, clearly. But with all of the different comparisons that they did using different land-based animals, different ocean-based animals, and different species of cephalopods, I was really impressed with the level of detail that they went into and the fact that their equations can be used again in the future. So they've said, this is a starting point. It's the most comprehensive study on brain development that we've got so far, but this isn't where we finish. we can keep going with this, and these equations that we've created will be helpful in doing so. So yeah, from a scientific perspective, that's a pretty good thing. If you are someone who is very interested in cephalopod cognition and brain development and understand mathematics, go check out this article. It is really interesting, but it's got a lot of difficult concepts.
And I'll certainly link to it below. I have to ask which, because as you know, Aaron, when we go over cephalopod news and there is a discovery or a paper, it will be picked up by multiple publications. So the version that I'm looking at, I think, is a much more watered down version. I don't want to undersell it, but I don't think, and I think it links to the paper. Is this from iScience? Is that the one that you're reading from?
I have the iScience.
Got it. I'm looking at and it is referenced from this Nautilus. So nautil.us is the website. I'll link to both below, but that's the one that I was looking at. And it was basically talking about shallow water cephalopods developing bigger brains, right? To your very point about now you got into the science of how they kind of arrived at this. So I definitely will check out. And again, I'll link to them both. So you can all at home go full nerd mode, or you could go, kind of, a little more on the surface. But
that's the problem. When I see these, I go full nerd mode and I go to the source material and I'm like, okay, I've got to figure this out.
Dr. Aaron Boyd Evans, I wouldn't expect anything less. But yeah, so the idea that they develop larger brains in shallow water, but they would not attribute it to social interaction. It's kind of interesting, right? Because and you alluded to it yourself, being like, yeah, okay, but still, it all goes together, doesn't it? I mean, like, so you can imagine in shallow water, certainly the substrate is more interesting. There are more kind of like micro interactions that an octopus will have there versus like a more deep sea benthic octopus. Presumably, and I'm making presumptions here, and I don't know. First of all, I'm not really all that good at math, and I don't know what they put together that you read that I have not. So I'll check that out too. It's definitely very interesting, and it is cool, as you underscored, that is reusable, and this is kind of a starting point. So that's an important story. Appreciate you bringing it to the podcast here this month, Aaron. Good one. And I think so those are the stories. We blew through them, and I have the one bonus one. So in every Octopodcast, Aaron and I like to kind of scan and understand where across this great land called Earth that any aquarium is newly housing any cephalopods. And we have one in the Baltimore Aquarium on July 9th. There was an article by WBAL in Baltimore. So Tilly, which is short for Octillery, which is a Pokemon character.
Yes.
Is a giant Pacific octopus who has been added to the National Aquarium in Baltimore, which is an amazing aquarium. If you're anywhere near it, anywhere on the East Coast, it's totally worth the trip. Amazing shark tanks and everything, but always, almost always carrying an octopus or a cephalopod. So now they have a giant Pacific octopus, which is always great to see at the beautiful Baltimore Aquarium. Aquarist Tom Studer, if I'm pronouncing that correct, said Tilly is incredibly intelligent. She's cool. She's so engaging. She's actually responded visually to different people, which is, think about that. That's imagine going to the aquarium, you get that season pass, and it's like Remarkably Bright Creatures, right? I mean, I think the movie I didn't watch, and they're talking about a lot in the article here, of course. But yeah, very engaging octopus. There are no gaps for her to get out. Always very important. Let's see. And they're just talking about her body, like, the only hard part of her entire body is her beak, which is about 1.5 inches. So she weighs about 26 pounds. And she does not like clams, but she likes the shrimp heads. So they leave the heads on the shrimp because that's what she prefers. So interesting stuff. Go check out Tilly the octopus at the National Aquarium in Baltimore if you can. All right, I think those are our stories for this month, Aaron. I think that's a pretty good set. Anything else from your end?
No, I'm so happy that we waited that extra week because I think we got some really good kind of bonus material to talk about this month.
We did. And, if anybody wants more, of course, the Octobot every Saturday posts a new edition covering cephalopod news that it has scraped up through the legacy RSS newsfeed. But now, with the help of AI, Octobot is now distilling the top stories, and I work with it every Saturday, and make sure that it puts something worthwhile of the TONMO audience. So do check that out every Saturday, and that's broadly what Aaron and I draw from. Although there's sometimes other sources that we pull from as well. And there was a lot that we left on the table, actually. So there's a lot going on in our forums. Aaron predominantly manages our species set of forums, the family and species forums. There's some interesting activity there, lots of interesting footage being contributed by our members, which is super cool. So like and subscribe to this YouTube channel, and then definitely come over to TONMO if you don't have an account. Sign up; it's free, it's fun, and there's like 26 years of cephalopod information there, and conversation threads and whatnot. So lots for you to do there too. All right, cool. Aaron, we'll do this again next month. Thanks for joining.
See you later, everyone.